also contribute to nitrosamines. So finished products which are packed in blister packs which have nitrocellulose, they also contribute to formation of nitrosamines in the product. And as I said, pH of the formulation is critical, both in API and formulation is critical. So when we talk about controlling the nitrosamine in the drug product, we need to, as a formulation person, a formulation scientist would always give a preference that the API should be free of amines, but that is not possible. So what we need to do is, we need to cut down the source of nitrites and nitrates. Since in the view of time, I'll move to important part. Acceptable intake limits, I think what we need to understand is that all the nitrosamines are not carcinogenic also. The size of the NDSRI, the nature of the substitution can play a role in determining whether that NDSRI is carcinogenic or not. And in most of the cases, the surrogate approach is used. The Tanimoto score is used, wherein what we do is, if there is any new nitrosamine or a novel nitrosamine, we try to find out a molecule with a structural similarity to that nitrosamine for whom there is some toxicity assessment available. And based on that, the acceptable intake is proposed. And in terms of safety and toxicity assessment, in silico study with direct access and SARA-based database kind of softwares are available which provide some toxicity assessment. But Ames test is something which is not recommended by the FDA, and in vivo assays, transgenic assays, bacterial mutagenicity assays is something which FDA expects to prove that the product or the API is free of nitrosamine toxicity. So the industry is dependent on the surrogate methodology and matching up novel nitrosamines with structurally similar nitrosamine that have already been tested in rodent carcinogenicity assays. So I'll quickly go through the main points which I want to speak about today. Where can academia step in and support the industry, and where do we need an academia-industry collaboration? First, to start with at the API level. As every API is different, so the methods which are related to each NDSRI is distinctive and it takes time and resource to develop. Academia can focus research on development of further improved analytical methods for NDSRIs, methods that are more rapid with improved screening, methods that are not overly cost prohibitive and which can be adopted easily. Currently, industry, we are heavily relied upon LCMS and GCMS techniques for analysis of nitrosamine impurities. These techniques are important during the confirmation of the presence of a particular nitrosamine, but these are again costly, these are not easily available, and they can also give false positives due to the presence of artifacts arising from the formulation. Academia can work to identify alternative analytical methods which are fast, which are reasonably priced and common, and that can be easily used on a regular basis for NDSRIs which need to be checked on release and stability. It's not an easy task, but this is one area where I strongly feel if academia can put in more resources, more time, because industry is busy handling the current workload which is there. The current burden which has come or whatever due to the alarm which has come due to the nitrosamines, industry is busy handling the routine products with already commercialized products. So if academia can focus on developing good analytical methods, here's an opportunity for us to collaborate. Secondly, at the drug product level, fast, affordable, and precise determination of nitrite and nitrate in reagents is critical. And this may be considered as the first step towards understanding the risk of nitrosamines. So, academia can support by identifying the root cause of nitrites in excipients or developing commercial process for manufacturing nitrite-free excipients. Currently, some suppliers for microcrystalline cellulose, for example, have changed their processes to come out with nitrite-free, or it's not nitrite-free, it's low amount of nitrite. But if we can work out and spend some more time into basic research as to why these excipients have such high level of nitrate and can we cut down the source of nitrites, this will help resolving the nitrosamine issues in the formulations. If we can develop analytical methods for testing nitrites and nitrates in any such excipient where there is a possibility of presence of nitrite and nitrate, that could be another contributing step. And thirdly, one of the most important help which can be done by academia in the formulation is if we can evaluate new nitrite scavengers which are acceptable as per IID. There are nitrite scavengers reported in academia, many, but only ascorbic acid and alpha tocopherol are currently acceptable academia can do some research, come up with more nitrate scavengers which are acceptable as per ID, and we do not interfere with the bioavailability of formulations. There is a great scope to contribute to numerous products and resolve the nitrosamine concern in the formulations. Then, in terms of setting the acceptable intake limits, if academia can support through the in silico screening for checking the mutagenic potential of impurities, or support research on alternate in vivo assays that can be considered adequate to qualify an NDSRI. The target is definitely speed within decent cost. Academia can also assist to justify the approach strengthened by the scientific basis of molecular weight correction, which currently EMA and Health Canada have not accepted. FDA is also silent on that. But if we could work out some scientific rationale on this or come up with an alternate approach to predict the acceptable limit of NDSRIs based on a surrogate method, that would be really helpful. And one of the challenges for the industry currently is to obtain suitable reference standards for nitrosamines being tested. So academia could explore this area also to proactively support with the availability of nitrosamine impurities or reference standards for the APIs which are secondary tertiary amines. This could be helpful, and even if it is not at a GLP level, it's just a preliminary work at exploratory level or non-GLP levels, that can also be a part of collaboration to get an early indication and a quick screening. So this is my last slide. I'll just conclude. In terms of the risk assessment, I have talked about the API needs to be thoroughly screened to understand the role of all the raw starting materials, intermediate solvents and everything. Next important step in terms of finished product is to cut down the source of nitrites and nitrates. Excipients containing nitrites and nitrates like MCC, previous starch, SSG, povidone needs to be screened diligently. Alkalizers have been helpful to achieve nitrosamine control. Antioxidants like ascorbic acid are acting like nitrate scavengers. We need much more than ascorbic acid. For NDSRIs, strategy should be to find out the appropriate surrogate to read through study and justify the ADI in wider ranges. So I will conclude by the statement that nitrosamine controlled drug formulations are possible to achieve with very low and acceptable quantities. If we do a timely risk assessment, if we have a better understanding of the mechanism of formation, if we have more powerful analytical methods, if we timely execute the control strategies, and all of the above can be facilitated by an active academia industry collaboration. Thank you.
Thank you very much, Dr. Madan. The problem is huge in the industry, so questions?
What you mentioned is, I think, really a very alarming, and specifically if it's coming from a very common excipient which we are using in formulation, right? So is that these excipients also have some standards now on nitrate contents, like, because most of them are like pharmacopoeial standards, right? So is there any controls coming from there also on their standardizations, or we have to do all the,
right. In USP, nitrosamines appeared for the first time just 3 years back in 2019, and then it got implemented in September 2020. In European Pharmacopoeia, again, it got published for the first time in late 2019 and got implemented in 2021. In IP also, general chapter, it has been introduced just now in 2 months back, although there is no date of implementation. But there is a guidance in general chapters of all the pharmacopoeias now, just 2 years back these have been introduced, which talk about all these risk assessment and everything. And there is a basic guidance as to how we need to proceed with the risk identification, the risk assessment at API stage, at the formulation stage, and all the excipient stage. Now, when we come to excipient by excipient, currently there are no pharmacopoeial limits. But the industry is sensitized, the excipient manufacturers are sensitized. If you talk to any excipient manufacturer or MCC manufacturer, he is aware that what is the level of nitrite or nitrate which is expected. Currently, they don't have the controls. A lot of work is needed for that.
So even they don't have like kind of current control limits for them, right?
They have the limits, but we need to go much below. For example, I'll just share with you. For example, Rocket provides MCC, DuPont provides MCC. The limits are around 4.5 ppm. I'm talking about ppm, but 4.5 ppm is also high. We need to talk about ppb limits now, because otherwise controlling, if any traces of nitrates and nitrates are there in the excipients, they react with the amines. And you look at the APIs which are already approved worldwide till now. Many have secondary amines, many have tertiary amines, many have quaternary amines. So API you can't stop now. These are the APIs we have. Right. In terms of excipients, we have to cut down the nitrates and the nitrates.
Thanks, Sumit. I mean, it was really very important topic, and you touched base on. Thank you for that insight.
Yeah, there's one quick question and quick answer.
So very good presentations, Dr. Madan. So we are also working, being academia or Institute of National Importance in Naipur, Ahmedabad. We are working from last couple of years. And we have almost covered whatever you were talking about on sensitive methods. In fact, we will be having ISO as well as NABL accredited lab for that. So quick question I have for your presentations. You are talking about nitrates and nitrate both, but why nitrate? Because generally nitrate cannot convert to a nitrite to do a nitroso.
I think I did, here's my presentation. Because nitrate can convert. So and it can, under the acidic conditions, it may form nitrous oxide, and then it may react.
Okay, means I will try to catch up later, but because nitrate converts to nitrite, it generally are reductions, and which can happen only if there is a metallic impurities and other things are present, but otherwise they are not there.
Right, but so, nitrate can then convert into nitrous oxide, and then it may...
Okay. And another thing what we are doing currently is in generating an in situ NDSRI. So we have a protocol developed for any of the molecule because the standards are not available. So we also get a lot of queries from different companies like Sendos and a lot of other companies. So they are also not getting NDSRI standards.
They are
and
also not getting
standards. So what we have generated is we are generating an in-house in situ generation of NDSRI and trying to do a weight percent or a RRF based on the inverse corona CAD and advanced analytical technique. But is it acceptable where the standards having a RRF means whatever we are generating is not 100% pure?
As I said, it will help us in the early screening.
Makes
sense.
It
will give us a quick indication. Currently what we are doing is whenever there's an alarm or there's a new nitrosamine or a novel nitrosamine with which we become aware, we need to first prepare those standards and everything. If we can get support, so that will help us in an early screening and quick indication whether there is some risk or we are.
Yeah, means we are supporting some of the industries.
I think we can discuss it later. And Dr. Sumit Madan has given a very extensive overview, and we would like, on behalf of the organizing committee, we would like to recognize him.
We move over to the next speaker now, Mr. Ashish Gogia. Mr. Ashish Gogia is Executive Vice President, R&D at Medrich Meiji, Bengaluru. He has worked in several leading pharmaceutical organizations in the product development and research department, like Ranbaxy Research Labs, Aurobindo Pharma, MacLeod's Pharma, Cadila Pharmaceuticals. He is a master's in pharmacy from Delhi University, and today would be talking on a perspective from the CDMO side on creating portfolio of complex enabling technologies in CDMO environment. Over to you, Mr. Gogia.
Thank you, Doctor. Very good afternoon to all of you. And first of all, I would like to extend my gratitude and thanks to NIPER Organizing Committee for inviting me and representing our views. So on behalf of Medrich and Meiji, I would like to first take a few minutes to introduce my group. And as the previous speaker mentioned, I may be going for dumping of many slides over here, so please don't get scared about it. It is just to showcase you the emblem, the kind of technological words we feel, and we are going to show the delivery of the drugs which are going to happen in the future. Currently, our span is for generic drugs, what we do here, but we are aspiring to become one of the delivery house in India, Bangalore, by not only conceptualizing the product at our R&D, converting those thought process from R&D, making batches, to our clinical batch manufacturing site in Bengaluru, and then post-approval, those products should reach out to the market which are based in Japan as our primary market, followed by Europe, Canada, Australia, New Zealand. At the moment, we are not much into the US, so I will be restricting my discussion towards the group which I represent, and I'll be little more on the fast-forward manner, keeping these time constraints about it, so please forgive me for that part. So Medric is a part of a major pharma giant in Japan called Meiji Group, and we represent in India their CMO and CDMO arm. We do the business with the major clients, and this association happened in the year 2015. Now in 2022, we are 47-old existence in India, but now we known as the group of Meiji. This is the group we are talking. It is known for more chocolates, confectionery, dairy products, and we are the part of ethical pharmaceutical group. Meiji Seika Pharma Company pharmaceutical segment. In this segment, they have two arms. One, they call it as their innovation pipeline. Another is called as the generic segment. So the generic segment is now the Medric, which takes care of this operation from India. This is the global map, what we operate. We are based here in Bangalore, but we do the business globally through our subsidiaries here. And I'm particularly talking about the space, how the CDMO companies operate. We don't have our own products to sell to the customer. However, we provide services to many, many pharma clients for producing the cutting-edge technology drug deliveries options from these products. In total, we have the regulatory approvals from UK, Australia, Canada, Romania, and also we have included for PMDA Japan. We have close to 220 scientists engaged at this center in Bangalore. We have 30 officials in regulatory affairs, and we primarily focus on the oral solid dosage forms. This slide is more like showing the engagement that year on year, or I should say month on month, every month, how many studies go through our clinical bioequivalence study programs, and literally it is every week one product is going. So which makes around 36 to 48 products first time developed in our R&D center, going through the clinical batch manufacturing, and then putting it across for the registration market. So this is just a track record to showcase that engagement. These are the photographs of our center. I am based here at Bengaluru in R&D, whereas all our GMP operations happen through these sites. And this site is particularly dedicated for our Japan portfolio of projects, which gets manufactured, then supplied to Japan market as our unique capabilities toward it. Now, coming a little bit out of Medric and explaining about the CDMO scenario, so I thought it is better to give me a context here that India is a hub which does a lot of exports in the pharmaceuticals, but in terms of the revenues or the valuation, we hardly get in the ratio what we expect from the volume. So the volume speaks the language, but the values doesn't speak the language. Why? Probably this is the reason which we got it from it, and this was our broad understanding. We mostly operate from India on intermediate and bulk, commodity generics, conventional dosage form, and maybe this decade of 2020 onwards, we are seeing a trends happening of newer and complex formulations going as a value-added and branded generics. The future definitely lies if India can have the capability to be associated with NIPER and in fact brainstorm towards it, how we can escalate this kind of a capabilities in India and produce more and more into the higher margins part. Imagine if with the same volumes, keeping this kind of a margin, the effect on economy, the effect of products which we'll be making will be an outstanding performance from the group here. So my talks will be mostly covering on the topic which we make is how many complex formulations we have done so far historically. So if I see from the last 6, 7 years and what we are going to do in the next 5 to 10 years, this is the emblem through a case studies model and presenting the actual live products, what we are developing, what we have developed, what we are filing or commercialized. I thought it is best to explain to my fellow scientists who are in this field and would be prospering to come and do in the similar environment of developing more and more high-tech enabled products. So the first case study which I would like to put across is the multi-unit particulate system. In India, my colleague Sumit and the earlier fellows have explained, these are the big companies who do a lot of research, commercialization. They have their own capabilities to do multi-unit particulate system dosage form, particularly for export to the regulated market like Japan, US, and Europe. But what about others? They look for such kind of an opportunities. Can we make an affordable dosage form by putting across a GMP or a R&D site and put these ideas into the action, even if they don't have that kind of a capabilities of their own? So CDMOs become a definitive answer in such situation. This is the product I'm naming here is Europidil, which was developed, started somewhere in 2010. And now in 2022, it has seen the light of a full-blown commercial at our site in Bengaluru. And it has gone through a lot of stages of complexities and all, which you will be able to see here. Initially, it was originated in 3 years, we should be able to commercialize it, but it took 8 to 10 years to reach to this level. So the brand name is Ebrantel, 3 strains. It is available in Europe. Not much literature is available. We were asked to do the reverse engineering. These are the major excipients which control the drug product. Pellets are a population. These are all white colored in a capsule, but we don't know which percentage of pellets are of what release, how many immediate, how many sustained, how many modified release. So we did this study. These are the 5 major performances. Initial 2 attempts failed. Next 3 we passed, and that's how this product got commercialized, and we are now making this product. But the complexity never resides only on solving the product in development or in the beginning. It starts when you are in the commercial mode. The batches were taken in the development, maximum 58 kgs. Dossier got approved from the Europe. Now the company is making today a batch of 1.3 tons, and we have been asked to produce such batches, minimum 45 to 50 in a year. Any equipment available which is commercially viable facility, they cannot produce more than 12 batches a year depending on the complex cycle of this product. So this is the kind of complexity we deal in oral solid, and now we are making efforts to further make more and more smarter way of producing these kind of products in a pellets. Another angle of complexity here is proving to the agency when you have done the bioequivalence studies, the 5 studies plan which I have shown you, the moment you change the manufacturing site, the moment you change your scaling up from 58 kgs to 300 kg and then 300 kgs to 1.4 ton, they ask for repeat bio studies. How many studies you will do? What are the possibility of getting those approvals? So we took the approvals through IVIVC model here, and the registration was granted through this successful complexity model, and now we are in the happy space of commercialization. Some cases we resolve, it took time, but some cases we have just started the journey where we are still not able to resolve, and we'll be looking forward to the NIPER and Professor Bansal and his lab and many more experts in the academia to tell us how to solve this kind of a problem. So this is the second product, barnidipine. It is well there in the market, no competitor. People are asking us to develop the product. We could not see the light of the day of this formulation due to this reason. 99% of the barnidipine goes out of the system without even absorbing. The absorbed drug is only 1% of barnidipine, and within that 1% drug, it is so highly variable, 55% of the variability built in the compound. Sometimes it shows bioavailable, sometimes it doesn't show the bioavailable. How to get the approval of such kind of a product? So the study in fasting shows the drug product we developed is faster. In the fed, it shows we are drastically poorly bioavailable into it. So converting such opportunities, further brainstorming, putting the investments, and resolving is the future, something which will be depending upon taking the expertise from NIPER and trying to convert them into the commercial space here. Third important MUPS case is the MUPS ODT dosage form. This, according to our assessment, is a very, very niche area. Even though these many 11 MUPS ODT products are available in the market, no Indian player other than the top 3 or top 5 players are able to export such kind of formulations to Japan, to Europe, to US. They are restricting themselves to make such kind of a products in a capsule dosage form and exporting it, but not in a tablet form. Obviously, there must be some barrier. Obviously, there is a complexity. So this is the complexity. When you have to produce a tablet which contains 2 distinct particles, one which is a very, very heavy particle like a pellet, the other one is a powder, mix them into the tablet and prove to the regulatory agencies through all the yardstick, is going to be challenging. So this is the typical diagram how we have been conceiving this product. It starts with the small pellet. The red one is the drug which gets loaded, then start intermediate, enteric, different enteric, blend them, compress them, and which should give a right kind of a performance. And if you look at it at the typical composition, in our terminology, we call it as Gola technology. Gola means it is so many goal rounds here and the prefaces that you have to keep optimizing what it contains at each Gola layer to get the desired output of the MUPS tablet. But the problem doesn't happen if you have successfully designed a good formulation of MUPS tablet. The problem doesn't finish if you have a good impurity control. The problem is if you have to produce 2 tons of a tablet of a MUPS, how are you going to commercialize it? This product has a tremendous problem in the segregations. So this is what we have observed. And we came up with the answer like this. We were going for $1 million investment in putting a plant which will produce day in and day out, putting the international machine with a single hopper to produce it through gravity feeding. And we said, no, this cannot be the answer. The answer lies in these kind of machines which are still to take place in Indian pharma industry. Very few players, but Medric is the one which has invested now into this kind of capabilities. The second broad category of the complex technologies which we are imbibing at our place is we call it in a drug delivery system, and which is a disruptive technology. It is more than 30-year-old technology, but it's still no replicas, no generic players, no CDMO services. How to get an Oros product into any of these technologies called mono, bi, or tri-layer. This is Adalat product, very famous nifedipine. We are now the biggest manufacturer of nifedipine, laser drilled at our site, developed at R&D in a non-GMP environment, meaning having a laser drilling capability on the product at R&D site, followed by a mid-segment in a GMP, in a clinical trial segment, and post-approval on a commercial scale. So these kind of investments on drug delivery is only to facilitate that we enter into providing affordable but high-tech healthcare product for the industry. So this is the actual machine which is lying in our GMP location, and these are the typical programs. You have a bilayer tablet, the machine will segregate the drug layer containing the coated one, the colored one, and the below is the placebo layer, so it will flip, and only the laser drilling will happen on the drug portion. It will not happen on the placebo portion. And this tablet will come out and will produce a 24-hour drug release. This is the benchmarking we have done through the studies on the size of the laser drilling hole. And these are the bioequivalence studies. Initial failure, but we went on believing the capabilities of the group that yes, we will be able to achieve it if we are on a good scientific path. And now we are there in the bioequivalence meeting the criteria, and dossier is filed. We have another interesting product coming in the same laser drilling system, but it's a tri-layer tablet. You not only have to produce bi, but this time it is going to be a tri-layer and still a laser drilling system. We have an opportunity in a mono-layer tablet, which is a laser drilling, where the patent is only on the side of the tablet. It is not on the center. So they have created a release pattern of this product by drilling on the sides of it, and we have been asked to produce a formulation which is bioequivalent, but not following the patent because the patent is valid till another 7 to 10 years. Third option, very quickly, I wanted to put it, which is the industry